Yes, so very good evening to all my students. Today I'm going to start with biotechnology principles and processes, and that is I'm going to start with the topic called PBR 322. Classmate PBR 322. Clear, Babu? So आज ये टॉपिक तो है about classmate PBR 322. Fine, it's a very, very important topic. So whatever information we had in the very last class, let us start from here. Otherwise, problem ho jabe bujhte. So let us start with that. Okay. So what we have completed about biotechnology in a nutshell. Fine, in a nutshell. Chalo. So bio means what? Bio means life. Science, technology means science. So basically we are going to use what kind of life for our human benefits, human mankind. We are going to use bacteria, a prokaryotic cell called bacteria. And in bacteria, what is so very special about bacteria? They have got extra chromosomal DNA, which is called as plasmid. They have got extra chromosomal DNA, which is called as plasmid. So संग में क्या है? They have got a circular DNA, that is their bacterial chromosome. Fine. Now this plasmid, we can take this plasmid out from the bacterial cell. So there are process, we have talked about the process how we can take out the plasmid from the bacterial cell. The moment we are going to take out this plasmid, we can use this plasmid as a vector. We can use this plasmid as vector. Now what is called vector? Vector, we know that for malaria, what is the vector? For the disease called malaria, what is the vector? The female Anopheles mosquito is a vector. So female Anopheles mosquito never gets malaria. Fine, it only carries what? Anopheles. So it carries the agent, it carries the causative agent called plasmodium. Just like that, this bacterial plasmid, it acts as a vector. What we can do with this vector, we can introduce our gene of interest inside this vector called plasmid. Got the point? And what is our gene of interest? We have talked about one of the example in the last very class is insulin. So human insulin, the particular part of the DNA, which can give rise to insulin, what we can do is we can cut this insulin or gene of interest. This is our gene of interest. We can cut this gene of interest by the help of restriction endonuclease enzyme, by the help of REN. So they are the group of enzymes which can cut DNA at specific site. And what are the specific site? We know they are called palindromic site. Palindromic site is what? We know palindromic site is like this. One of the example we have talked about, GAATTC, 5' to 3', and from 5' to 3', if the bases are like this, GAATTC. Fine. So these are called palindromic sequence. So these are the restriction site where the endonuclease enzyme can cut the DNA. Over here, if the example of this one of the REN is ECOR1 called Escherichia coli RY13-1. I think we have talked about this, how to name it. E for Escherichia, CO for coli, R for the type of strain called RY13. So what is the type of the strain? RY13 is the type of the strain. And what is one? It is the first isolated enzyme from this bacteria. This ECOR1 is, it can cut DNA in between G and A, and can make what? And can make sticky ends. Just like we can, if the bone gets broken in this fashion, then the chances of this one to stick will be maximum if you compare it with blunt end like this. If the bone gets cut in this fashion, if you compare these two, this is what is called sticky end, and this one is called blunt end. This one is called blunt end. Just like that, this ECOR1 can cut the DNA and make sticky ends. Just like this, GAATTC, and over here GAATTC. See, this is what, after this ECOR1 is being utilized over here, ECOR1 is going to cut DNA in between G and A and can make sticky end. So in your book you can see one of them is the vector DNA and other one is the gene of interest. Other one is the gene of interest. बुक में देखो एक रेड में है और एक ब्लैक में है. Fine, and they are going to get stick. They are going to get stick. They are going to have double bond, and this is how they are going to get. And ultimately, what is going to happen if you introduce the gene of interest with the vector? What is going to form is recombinant DNA. What is going to form is recombinant DNA. So what is this? This is plasmid, and this is plasmid, and you have cut the plasmid. You have cut the plasmid by the help of ECOR1, and also you have cut this part of the DNA which can give rise to insulin, and what you have done is you have made recombinant DNA. And then you need to put this rDNA back inside bacteria. Now why you have chosen bacteria? Because bacteria is having a high reproduction rate. High reproduction rate. That means the part of the bacterial DNA where from the replication occurs is called ORI. And what is ORI stands for? ORI stands for origin of replication. ORI stands for origin of replication. Now what kind of DNA we human beings are having? We are having linear DNA. हमारे पास क्या किस तरह का DNA है? We have got linear DNA. But in case of bacteria, they have got circular DNA. So in case of circular DNA, they have got ORI. And from ORI, when they start replicating, ultimately what happens is, if there are two bangles, one of the bangle makes another bangle. So the two bangles are going to get attached like this. And there is a nick which is required to make it free. Just like that. This is how the bacterial replication also occurs. And the process is called catenation. The process is called catenation. Okay? Now, so what we have got rDNA. We need to put this rDNA back inside bacteria. And there are processes how we can put them back. Put the rDNA back inside bacteria. For that, what we need to do is, we need to make the bacteria competent. We need to make the host competent by culturing it in divalent cation. By culturing it inside divalent cation. That means we have to use calcium chloride solution, divalent Ca+2, where we are going to make the bacterial cell competent before introducing the rDNA inside it. And then what we need to do is, we can use 4 processes to introduce our gene of interest. And or to introduce rDNA inside this bacteria. So what are the 4 processes by which we can introduce this rDNA inside bacteria? So this is the gene of interest, which is insulin forming DNA. Shrijil, are you getting me? Now, what we need to do is, the first thing is, we need to use this process called microinjection. Lastly, we talked about that. We have talked about this, I think. Yes. Then we have to use gene gun. The next process is gene gun, which is also called as biolistics. Also called as biolistics, where we are going to wrap this rDNA. We are going to wrap this rDNA by the help of what? Gold or tungsten. So this is the gene of interest, and we need to wrap it by the help of gold, either gold, Au or W, tungsten. Okay? And then this is gene gun, and then we are going to bombard this inside the bacterial cell. And third is what? Heat shock. Third is heat shock. Heat shock. First inside 42 degrees Celsius, then 0 degrees Celsius, then back into 42 degrees Celsius. And the last one is by the help of disarmed pathogen. So we are going to take the bacteriophage and disarm it. What is the meaning of disarming it? So if this is the virus, we can take the genetic material from the virus. It can be either RNA or DNA, or sometimes both. So we have to take this out and then put our gene of interest inside it in lab condition. So this is the rDNA, and then we are going to allow this virus bacteriophage to infect a bacterial cell. So what it's going to do is, by the help of spike, it's going to perforate the cell wall and push this rDNA inside it. Push this rDNA inside the bacterial cell. These are the 4 techniques by which we can introduce our gene of interest, or in other ways, rDNA inside the bacterial cell back. Now the most important role is being done by this, what? Plasmid. Because plasmid is the vector. Plasmid is the vector. So what we are going to study today is about a plasmid which is called as PBr322. So PBr 322. This is a plasmid that today we are going to talk about it. Now what is PBr322? It's a plasmid. And this plasmid is being made by human being for our purpose. For our purpose. Now understand this. What is P stands for? P stands for plasmid. What is B stands for? B stands for the person named Bolivar, and R stands for Rodriguez. Rodriguez. Fine? Now, if this is the plasmid, which is there in your book, fine, a rough idea, this is the plasmid. This plasmid is having 4361 number of base pairs. 4361 number of base pairs. Clear? It's having more than 40 number of what? REN sites. REN sites. But in our book, we have got only 9 particular enzymes, 9 particular REN being shown in your book. And we are going to talk about each one of them. And then, the molecular weight is 2.83 into 10 to the 6 Dalton. So it's having high molecular weight. This is PBr322. Okay? Now, what is so very special about PBr322? If you see in the figure, in the book, you can see there is ORI. What is so very important over here? There is a ORI over here. There is a ORI. What is ORI? Origin of replication. So replication will start from here. Then there is ROP. ROP structure present over here. Let's make it like this. Fine. This is ORI. ORI. This is ROP. What is ROP? Who is going to tell me? ORI, you already know it. It is origin of replication. Origin of replication occurs over here. What is ROP? What is ROP? For replication, the proteins which are needed are coded from here. The proteins required for replication are the protein required for replication is being coded from here, ROP. Then you can see there are 2 structures like this. There are 2 structures like this. Can you see this? 2 of the pink structure present in your book. You can see that. One is ampicillin, AMPR, and you can see over here TET-R. What is AMPR? What is AMPR? It is called ampicillin resistant site. It is called ampicillin resistant site. Ampicillin resistant site. And what is also present over here? Tetracycline resistant site. Tetracycline, ampicillin, all of these 2 are what? These 2 are antibiotic. These 2 are antibiotic. So tetracycline resistant site. Tetracycline resistant site. Resistant site. Okay? Now, if you see in your book, in this ampicillin resistant site, can you see some of the REN being given? What is the name of the REN being given in ampicillin resistant site? One is PST1 and another one is PVU1. PST1, PST1, Roman 1, and PVU1, PVU1. Let me tell you what is PST. What is PST? You can easily make it out. Roman 1 means it's a REN. It's a type of REN. So how to name REN? Just like ECOR1, PST stands for PST1 stands for Providentia, Stuarti. It's the name of the bacteria, Stuarti, T-double-I. And what is 1? The first isolated enzyme from this bacteria. So it's a REN, it's a type of REN. What is the next one? PVU1. PVU1. What is PVU? It is Proteus vulgaris. Proteus vulgaris 1. What is the first isolated enzyme from this bacteria? And you can see in the book, what is present over here at the top? You can see ECOR1 and CLAV1 is present. ECOR1 and CLAV1 is present. Along with CLAV1, you can see HIN3. So ECOR1, you already know about ECOR1. You already know about CLAV. ECOR1 you know. Escherichia coli RY13 1, the first isolated enzyme. What is this CLAV1? What is this CLAV1? CLAV1. It is caryophanon. Caryophanon. Latum. Caryophanon latum. 1. First isolated enzyme. Is the name of the bacteria. What is HIN3? What is HIN3? The next one is HIN3. What is that? It is Haemophilus. Haemophilus influenzae. Haemophilus influenzae. D is the type of strain and third isolated enzyme from this bacteria. Fine? And after, this is HIN3. Over here, HIN3. And then what? It's present in tetracycline resistant site. You can see BAMH1 and SAL1. BAMH1 and SAL1. So over here, you can see BAMH1. And over here, you can see is SAL1. SAL1. So what is BAMH1? What is BAMH1? It is Bacillus amylofascians, amyloliquefacians. H is a type of strain and 1 is the first isolated enzyme from this bacteria. First isolated enzyme from this bacteria. Something wrong. No, you have to switch it off. Okay. Now, Bacillus amylofascians H1. Okay. Now BAMH1. What is the next one? SAL1. SAL1. What is SAL1? It is Streptomyces. It is Streptomyces. It is Streptomyces albus. Streptomyces albus. The Roman 1 is first isolated enzyme from this bacteria. All well? Okay, SAL1. Tell me what is the next one? What is present over here? Just after SAL1, you can see PVU2. PVU2. In the ROP. PVU2. Again, what is PVU? We have already done about that. PVU means Proteus vulgaris. Number 2. PVU2. Proteus vulgaris 2. Okay, Roman 2. Okay. So these are the different types of sites. Restriction enzymes. Now, see, there are two terms that you will be getting over here. One is ampicillin resistant and one is tetracycline resistant site. And these two are called as the selectable markers. They are called as the selectable markers. Selectable markers. Fine? Selectable markers. And what is that? One is AMPR and one is tetracycline R. Okay? Now understand. See, ampicillin and tetracycline, what is common in both of them? Both of them are antibiotic. That means they are not going to allow a life to grow. Antibiotics never allows. It is against life. A life to grow. That means, see, if our gene of interest cuts off this portion, let me draw it again. This is our PBR 322 and this is there are two sites. One is AMPR. Let's make it with yellow. Okay, this is AMPR. And let's make it with blue. What is it? Tetracycline resistant site. Okay. Now think about it. If our gene of interest, say for example insulin forming DNA. So we want to make insulin forming DNA, on a large scale. Last day we have talked about how after we have put this rDNA inside huge amount of bacteria, we have cultured bacteria in culture medium, made them grow up to billion of times, and then we have put them in bioreactor to get huge amount of insulin. Now by the help of this PBR322, what we can do is in a single reaction, we can make insulin, we can put our insulin forming DNA, We can put our insulin, we can insert our gene of interest inside this ampicillin resistant site. And also at the same time, if you want to make vitamin A from the carrot, we can cut out this part of the DNA and put it inside a tetracycline resistant site. So in a single step, we can get two results. Which one is going to be economical? Having only insulin or having both of them? Both of them. Got the point? So it's going to be cost effective for the company. Having bioreactors. Now think about it. So how on earth we are going to understand whether our gene of interest has gone inside ampicillin resistant site or not? How we are going to understand whether our vitamin A forming DNA has cut this portion and got inside this or not? Got the point? So there is a process how we are going to understand it. Is this clear? Dipti. Ampicillin resistant, this is tetracycline resistant. Sujila, clear? Now see. Now, there are three terms I'm going to use. The first one. Tell me, if I call this as a recombinant, another one is a non-recombinant. Non-recombinant. And the one is transformant. Transformant. Fine. There are three terms we are going to use. Which one we need to grow? Which type of bacteria we need to grow in a large scale? The recombinant variety, non-recombinant variety, or transformant variety? Transformant variety. Now what is the meaning of recombinant variety? Having our gene of interest or not? Yes. What about non-recombinant? So that plasmid is neither having insulin forming DNA nor it's having vitamin A forming DNA. So that is not of any use for us. So we are not going to grow those kind of bacteria. But how to understand? How we are going to understand which part of the DNA has gone, which part of the selectoral marker or site is having our gene of interest. So to understand, to differentiate between these three, it's a cumbersome process. In your NCERT, this is the term they have given. It's a cumbersome process to understand. Why it is a cumbersome? Because it is a difficult process. Now, why it's a difficult process to understand? Now, let's get back over here. Say for example, if insulin forming DNA, Sijila, you have to tell me, if insulin forming DNA cuts off this ampicillin site, say this ampicillin site is not there. Is not there. Ampicillin site has come out, ampicillin resistant site has come out, and our gene of interest, this blue gene, has cut off this particular ampicillin site and got inside it. Clear? Now this has become what for us? Tell me the term. Recombinant DNA for us. But what we want only one insulin forming DNA to be present over here, or we want both of our DNA to be present? Both. So how to understand that which part of our gene of interest has gone where? See, in this rDNA, we have to put this back inside bacteria, and if we culture this bacteria in a medium, say for example this is a plasmid, this is the plasmid, this is the plasmid, and this is our gene of interest which has gone over here. You tell me, if this is the bacteria and if we take this medium, we are culturing this bacteria in a medium having ampicillin as antibiotic. So the culture medium, in the culture medium I have put ampicillin as an antibiotic. Do you think this bacteria is going to grow? Or is not going to grow? Not going to grow. Tell me why. Because what is not present over here? Ampicillin resistant site has been cut off. Ampicillin resistant site is not present over here. So resistance nahi hai. If there is no resistance, it's going to grow or it's not going to grow? Not going to grow. So if it grows, then we can have an idea that our gene of interest has cut off the ampicillin resistant site and gone inside. Got the point? Got the point? Now, say for example, this is a master plate where bacteria is present. Bacteria is present in a master plate. So we have taken this portion of the bacteria and we have cultured this. And found that this portion of the bacteria is not growing in ampicillin medium. That means this portion of the bacteria is having some of our gene of interest inside it. Is this clear? Any issues about this? What is master plate? বাড়িতে দই বানিয়েছে মাস্টার দই প্লেট সেখান থেকে অল্পটুকু কেটে নিয়ে এখানে কালচার করেছি নাও আন্ডারস্ট্যান্ড if this grows that means this particular bacteria is going to grow in ampicillin resistance ampicillin nutrient medium. The nutrient medium having ampicillin, the bacteria is going to grow or is not going to grow? It's going to grow. But if you keep the same bacteria, if you keep, to confirm it, if you keep this, place this same bacteria in tetracycline medium, tell me what is going to happen? It will grow. Why? Because it is already having what? Intact. Tetracycline resistant site is already present. Do you think our purpose has been solved? What was our purpose? To have both of them. To have both of them. Got the idea? So how many times we have to grow them? How many nutrient medium we have to use? Two. First, we have taken only ampicillin. Then, to reconfirm it, we have taken the same bacteria, allowed the bacteria to grow in tetracycline medium, and then found that it's not growing over here. It's not growing in tetracycline medium. Not growing or it's growing?
Growing.
It's growing. Why? Because it's having tetracycline medium. It is having tetracycline site intact. This is resistance. Resistance means what? Resistance means it's going to oppose. The tetracycline resistance এখানে কি আছে? এখানে intact আছে. That means tetracycline medium দিলে resistance টা আটকাবে. So কি করবে? এটা মারবে না গ্রো করতে হেল্প করবে? তাহলে যেটা এখানে গ্রো করছে না, it's not growing here, but it's growing here. It's not growing here, but growing here. That means our gene of interest has got inside where? In the ampicillin resistant site. See, inactivating ampicillin resistant site. Come on. Inactivating ampicillin resistant site by insertion of our gene of interest. Again, I repeat. Inactivating the ampicillin resistant site by inserting our gene of interest is called as insertional inactivation. It's there in your book in the next page. So what is the term? What is the meaning of this term? Insertional inactivation. Insertional inactivation. Insertional inactivation. What you are inserting and what you are inactivating. This is the plasmid again, and this portion is ampicillin resistant site. This is tetracycline resistant site. You have cut off this portion. Ampicillin resistant site has been cut off, and our gene of interest got inserted in this site. So our gene of interest has inactivated what? Ampicillin resistant site. And this is what is called insertional inactivation. किसको insert करने के बाद किसको inactive किया? Our gene of interest got inserted and inactivated what? The ampicillin resistant site. This is good for you, bad for you? Come on. That's what you required. That's what you wanted. This is called insertional inactivation. See over there in the page 200 in the old book, page 170. Page 170. Insertional inactivation. Clear? Now, what do you want? You want the vitamin A forming DNA to get inside tetracycline resistance. So, it's a transformed one. It's a transformant. But you want what kind of transformant? Having both the gene of interest inside, one in ampicillin resistant site and the one in tetracycline resistant site. To understand that if another DNA, another gene of interest has cut off and got inserted and inactivated tetracycline resistant site. Say for example, this is vitamin A forming DNA. Vitamin A forming DNA. And it has cut off this tetracycline. If it has made insertion inactivation in tetracycline medium. Adrito, tell me, if we culture this part of the bacteria, say for example, remember the master one. If you take this part of the bacteria from the master plate and check it and put this part of the bacteria in a medium having, this is the medium, and we have taken ampicillin and tetracycline, both antibiotics. So in the medium, we have taken both of these antibiotics. What kind of bacteria you want? A bacteria which should grow in this, or bacteria which should not grow in this? Should not grow in this. Because what you want no resistant site to become activated. So if the bacteria grows in these two conditions, a medium having both of them as ampicillin as well as tetracycline medium, if the bacteria grows, this one that you require to culture in a more amount so that you can keep those in bioreactor and have both of the product at the same time, insulin as well as vitamin A1. So how many times you have cultured them? 3 times. First you have put it in ampicillin medium, second you have put the same in tetracycline medium, third you have taken both of them and allowed those bacteria, some of the bacteria to grow in the medium having both of them, ampicillin as well as tetracycline medium. So definitely it's a cumbersome process, no? It's a difficult process, it's a cumbersome process. It's a cumbersome process. Any issues with this? Come on. So to separate from the recombinant from the non-recombinant, you have done this. Got the point? Now, what is the best way you can do it? There's an easy process how you can come out of this problem of culturing them in different medium. What you can do is, what is this site? Tetracycline site. What is this site? Let me see the original one. What is this site? Tetracycline site. What is this site? Ampicillin resistant site. We call these two as selective markers, or the cloning site, or the cloning site, because we are going to clone the DNA, the desired DNA over here, the gene of interest inside this. Now what we can do is, do you remember beta-gadosidase? Do you remember this enzyme?
Yes.
In lac operon? What was the name of that gene which produces beta-gadosidase enzyme? Z or Y or A? Z. So Z's gene, it produces what? Beta-gadosidase enzyme. A simplistic thing that what you can do is, you can take the Z gene and put it inside ampicillin resistant site. This is what you have taken. You have inserted Z gene over here, as well as in the tetracycline medium, you have inserted Z gene inside the tetracycline medium. Clear? Now understand. What is the utility of having Z gene in both the selective marker or both the cloning sites? What is the utility? The utility is, when you are culturing this bacteria, in the master plate, where is it? This is the master plate. This is the master plate. In this master plate, along with the nutritive medium, along with the lactose medium or whatever nutrient you have given the bacteria to grow, if you add chromogenic substrate, now what is the meaning of that? Come on. What is the meaning of the term chromo? Colored. Chromogenic. Genic means origin. जो color देता है. Chromogenic substrate. If you add chromogenic substrate in this master plate, if Z gene is there, intact, then it is going to give a characteristics blue color. Then it is going to give you a characteristics blue color. And the portion where Z gene is not there, it's going to make no color. It's going to be transparent. Now Adito, being the scientist, Adito is going to collect what kind of bacteria? Blue colored bacteria clones or transparent one? Sigila, if blue color is there, blue color is only going to form if Z gene is present. If Z gene is present. Z gene present means what? Z gene is present means what? It's present where? It is present over here. That means insertion and activation has not happened, has not occurred. Z gene है मतलब ये पोर्शन कट के नहीं निकला, मतलब जीन ऑफ इंटरेस्ट यहाँ पे घुसा नहीं, यहाँ पे भी Z gene है तो जीन ऑफ इंटरेस्ट यहाँ पे घुसा नहीं, अगर नहीं घुसा है यहाँ पे, if it is not being inserted in this, it's going to give you what kind of color? Blue. Are you going to take blue color and culture it? No. Are you going to make losses? Or you going to take only the transparent one? Now taking transparent colonies means it is having your gene of interest. Got the point? Any issues with this? This is about PBR 322. This is why you have taken this. What are the other types of plasmid artificially we have made for our benefits? We have made many of them. I'm giving two of them. One is PBR 325 and PBR 327. It's also being made by human. Got the point? How many sites being given to you in this book? 9 enzymes, but it can have more than 40 REN sites. More than 40 REN sites. Clear? So what is very important, what is the most important for you to understand about PBR 322 is all the examples being given, all the REN being given in your book. NCERT ने क्या नहीं लिखा? NCERT has not written the name of the enzymes, but you have to know those. CLAV1 caryophyllene latum, SAL1 Streptomyces albus, PVU Proteus vulgaris, BAMH1 Bacillus amyloliquefaciens. Fine. So PST Prodentia stuarti. You have to remember all of them. Clear, Babu? So this is about PBR 322. Any doubt? Let us check over here. Let us check over here. So what we have done this spooling process last day. We have talked about the spooling process, how DNA is being taken out. Fine. We have also talked about the type of nucleases enzyme, Sujila. We have talked about type of nucleases enzyme. The nuclease enzyme can be of two different types. What are they? Number one, exo and endo. Exo and endo. Exonucleus removes nucleotides from the ends of the DNA. Endonucleus cuts from within. From within. Fine. Yes. Nomenclature, how ECOR1 is being named. What is the name of the strain? RY13. ECOR means ECOR1. That means Escherichia coli RY13. Okay. Malayalam, Kanak. These are what? Palindromic words. Palindromic words. Geliotophoresis we have done. Ethidium bromide we have done it. What is the function of ethidium bromide? Staining technique. Okay. And then let us start with PCR. We have already done. We have completed this PCR. Yes. PBR. This is ORI. This is ORI. Okay. Now, let us start with this part. Cloning vector. Thermocytoticals we have done. The construction of the first recombinant DNA emerged from possibility of linking a gene encoding antibiotic resistance with a native plasmid autonomously replicating circular extrachromosomal DNA. What is the meaning of autonomously? Automatically, self. Of Salmonella typhimurium. Salmonella typhimurium. Cohen and Boer accomplished this in 1972. We have talked about Cohen and Boer. These two scientists are the persons behind biotechnology. 1972, by asserting the antibody resistance gene, cutting out a piece of DNA from a plasmid, which was responsible for conferring antibiotic resistance. The cutting of DNA at specific locations became possible with the discovery of the so-called molecular scissor. What is that? Restriction endonucleases are called molecular scissor, and molecular Glue is ligase. The cut piece of DNA was then linked with the plasmid DNA. The linking of the antibody resistance gene with the plasmid vector became possible with the enzyme DNA ligase, which act on the cut DNA molecules and joins their ends. The linking of antibody resistance gene with the plasmid vector became possible with enzyme DNA ligase. We have done it. This makes a new combination of circular autonomously replicating DNA created in vitro and is called as recombinant DNA. We have done this. We need to talk about PBR3 into origin of replication. Features required to facilitate cloning into a vector. This was the last day. So the origin of replication. What is origin of replication? This is a sequence from where the replication starts. And any piece of DNA when linked to this sequence can be made to replicate within the host cells. This sequence is also responsible for controlling the copy number of the linked DNA. So what is ORI? ORI can also think about the number of copy to be copied. So controlling the copy number of the linked DNA. So if you want to recover many copies of the target DNA, it should be cloned in a vector whose origins support high copy number. So what kind of ORI we are going to take which supports high copy number? When you make photocopies, what you do normally after you have put the original over there, you select how many copies you want. So the one which is going to support high copy number, that kind of ORI you are going to take. Plasmids are suitable vectors for gene cloning because they are small circular DNA with their own ORI site. Come on. Why you have taken plasmid as a suitable vector? What is the reason for that? Why you have taken plasmid? Because they have got very small circular DNA with their own ORI site. They have got their own ORI site. So what you can do is you can make some of them in lab conditions for your own benefits. Selectable marker. In addition to ORI, the vector requires a selectable marker, which helps in identifying and eliminating non-transformant. What is non-transformant? You require transformant or non-transformant? You require non-transformant. And eliminating non-transformant and selectively permitting the growth of the transformant. Selectively permitting the growth of? Is the non-transformant of any use to you? No. So what do you require? Transformants. Transformation is a procedure through which a piece of DNA is introduced in a host bacterium. Normally, the genes encoding resistance to antibiotics, such as, these are the name of the antibiotic, ampicillin, chloramphenicol, tetracycline, charamycin, etc., are considered useful selectable markers for E. coli bacteria. So what is the bacteria? We have performed all this in E. coli. In case of plant cell, what is the type of bacteria last day we talked about? What is the name of the bacteria? In case of plant cell, what is the name of the bacteria which we use for Agrobacterium tumefaciens? The normal E. coli cells do not carry resistance against any of these antibiotics. See, the normal E. coli cells do not carry resistance against any of these antibiotics. So you need to make it in lab. So if a recombinant DNA bearing gene for resistance to antibiotic, example ampicillin, is transferred into the E. coli, the host cells become transformed into ampicillin resistant cells. Become transformed into ampicillin resistant cells. The host cell becomes transformed into ampicillin resistant cells. If we spread the transformed cells on agar plates containing ampicillin only, transformants will grow, untransformant recipient cell will die. Right or wrong? Again I repeat. If you spread, now you have got what? Ampicillin resistant site. See, what you are doing, if you spread the transformed cells on agar plates containing ampicillin only. So what is the medium you have used? Agar. And what you have done? You have put it in ampicillin site, ampicillin nutrient medium. Transformants will grow, but untransformed will die. Untransformed will die. What is the meaning of untransformed? Having ampicillin site or not having ampicillin site? Since due to ampicillin resistance gene, one is able to select a transformed cell in the presence of ampicillin. The ampicillin resistance gene in this case is called as a selectable marker. So how many selectable marker is there in PBR 322? Two of them. One is ampicillin and one is tetracycline. Okay. Cloning site. See. Cloning site. To link the alien DNA, the vector needs to have very few, preferably single recognition sites for the commonly used restriction enzymes. Presence of more than one recognition site. Presence of more than one recognition site within the vector will generate several fragments which will complicate the gene cloning. Which will complicate the gene cloning, no issues, but it is going to be useful for you or harmful for you? Useful. E. coli cloning vector PBR322 showing restriction site HIN3, ECOR1, BAMH1, SAL1, PBU2, PST1, CLA1, ORI and antibody resistant genes, ampicillin R and tetracycline resistant site. ROP, very very important for your board exam. It's a very common question. ROP is a very common question for your board exam. What is ROP? Codes for proteins involved with the replication of the plasmid. So all the proteins which are needed for replication of this plasmid is encoded over here in ROP site. In ROP site. Okay. The ligation cutting of alien DNA is carried out at a restriction site present in one of the two antibody resistance gene. For example, we can ligate a foreign DNA at the BAMH1 site. See where is BAMH1 site? It is in tetracycline resistant site. So if you use the BAMH1 site, ultimately which portion is going to cut off? Tetracycline. So for example, we can ligate a foreign DNA at the BAMH1 site of tetracycline resistance gene in the vector PBR322. The technique of insertion of a desired gene into DNA of plasmid vector is called gene splicing. It's called gene splicing. You are splicing the gene with the help of DNA ligase. Foreign DNA fragment is joined. This is called splicing. This is called splicing. The recombinant plasmid will lose tetracycline resistance due to insertion of foreign DNA, but can still be selected out from non-recombinants ones by plating the transformant on tetracycline containing medium. Confusing? Got the point? Now, the transformants growing on ampicillin containing medium are then transformed on a medium containing
Tetracycline.
So first, where we have grown? Ampicillin. Second, you have taken the same bacteria, put it where?
Tetracycline.
Second process. Okay. And then, but the non-recombinants will grow on the medium containing both antibiotics. Now you have taken both ampicillin as well as tetracycline. So they are going to grow. They are going to grow. Now, in this case, one antibody resistance gene, ampicillin R, helps in selecting the transformants, and the other antibody resistance gene, tetracycline R, gets inactivated due to the insertion of alien DNA and helps in the selection of recombinants. In recombinant DNA technology, antibiotics are used to keep medium bacteria free. Medium bacteria free. Okay. Selection of recombinants due to inactivation of antibiotics is cumbersome process. It's cumbersome process because it requires simultaneous plating on two plates having different antibiotics. Therefore, alternative selectable markers have been developed which differentiate recombinants from non-recombinant. So how straight away you can differentiate between recombinant and non-recombinant? So there is a special way on the basis of their ability to produce color in the presence of chromogenic substrate. In the presence of chromogenic, this is very important, chromogenic substrate. The substrate which can give color. Okay, if it contains beta galactosidase enzyme. In this, a recombinant DNA is inserted within the coding sequence of an enzyme beta galactosidase. This results into inactivation of the enzyme, which is referred to as insertional inactivation. Star point, mind you, for your board and NEET. For your board and NEET, what is insertional inactivation? The presence of chromogenic substrate. The presence of chromogenic substrate gives blue-colored colonies. If the plasmid in the bacteria does not have an insert, what is insert? The gene of interest. What is insert? The gene of interest. So what kind of color it's going to give?
Blue.
Blue. Presence of insert results into insertional inactivation of the beta carotene. It is not going to be able to show its color, and the colonies do not produce any color, and these are identified as. So Advito is going to take blue colored ones or transparent ones.
Transparent.
Easily. So he has taken what? Recombinants or non-recombinants then? Only recombinants. Any issues with PBR need to come on? Nako? Very good. Vectors for cloning genes in plants. In case of plants, what is the vector you are going to use? Come on. Agrobacterium tumefaciens. A pathogen of several dicot plant is able to deliver a piece of DNA called YT. Tumefaciens DNA, tDNA. Transform normal plant cell into a tumor. What kind of disease it makes? Last day we talked about. Last day we have talked about this. Agrobacterium tumefaciens makes crown gall disease in apple. Crown gall, G-A-L-L. Gall means what? Tumor. So what kind of plasmid they have? TI plasmid. So what kind of plasmid they have got? TI plasmid. What is TI stands for? Tumor inducing. Tumor inducing plasmid. Clear? Now. And direct this tumor cell to produce the chemicals required by the pathogen. The tumor inducing TI plasmid of Agrobacterium tumefaciens has now been modified into cloning vector, which is no more pathogenic to the plants, but is still able to use the mechanics to deliver gene of interest into variety of plants. Think about, last day we have talked about vitamin golden rice. We have talked about golden rice. Remember? Golden rice. Golden rice is what? In Africa, people were suffering from xerophthalmia, night blindness, due to the deficiency of vitamin A. So, TI plasmid being taken, what was the gene of interest that we have put inside it? Vitamin A, and allowed this bacteria to infect the paddy field. E. coli and Agrobacterium are two bacteria which are most useful in genetic engineering. E. coli for animals, and for plants, Agrobacterium is used as a useful one for genetic engineering. Vector for animals. Vector for animals. Retroviruses animals have the ability to transform normal cells. What is retrovirus? Virus having RNA as genetic material. Have the ability to transform normal cells into cancerous cells. A better understanding of the art of delivering genes by pathogens in their eukaryotic host has generated knowledge to transform these tools of pathogens into useful vectors for delivering gene of interest to the humans. So once a gene or a DNA fragment has been ligated into a suitable vector, it is transferred into a bacterial plant or animal host where it multiplies. So we have talked about all this. Let us solve some questions. Transferring the recombinant DNA into the host. We have done it. Combinate host. Come on, last day we have talked about that. Divalent cation. So this is we have already done. So let us start with solve some of the questions. Extraction of the desired product. We have done it. Downstream processing. We have talked about that. Yes. How to get the finished product. Yes. Tell me, for transformation. Read out the question. For transformation. For transformation, microparticles coated with DNA to be bombarded with gene gun are made up of gold and tungsten. Very good. Come on, ballistics. Gene gun is suitable for constructing recombinant DNA by joining with vectors. In genetic engineering, the antibiotics are used. The antibiotics are used as
Selectable marker.
have a check. Have a check. Give a second thought.
Sir, option C.
In genetic engineering, the antibiotics are used. Only selective markers? Sure. What about to keep the cultures free of infection?
The infections.
Why not? Do you want any other bacteria to grow inside the master plate and make donsulin? So,
80% correct.
to keep the cultures free of infection. Clear? So that's all about
Duty question.
biotechnology. We have given some of the questions only. So in downstream processing, see, in downstream processing, what is required? Control, quality control testing for each product is also required. Fine. From the bioreactor, when the finished product has been made, till the time it reaches in your hand, quality testing purpose and all those stuff. Remember last day we talked about insulin was kept in vial and then class 1 preservative, class 2 preservative was used. Fine? So that is it. So that's all about this chapter, biotechnology principles and processes. So what you have to do is you have to solve questions. কি বলেছিলাম আমরা, what we have talked about, every day 25 questions, physics, chemistry 25 and biology 50 questions you have to solve. Fine? Next day what we're going to do, we're going to start with the next chapter, application. Biology principles and, biology and its applications. Very, very important. What are the topics we're going to study under that topic? First is BT cotton. Second, RNA interference. Third, Eli Lilly company. That is, we're going to talk about Eli Lilly company. The moment you open the question paper, you can see question number 26, 27, Eli Lilly company being given. Fine? That means humulin. Eli Lilly. E-L-I-L-I-L-Y. Eli Lilly company. Fine? Very good. And the fourth one is about gene therapy. These are the 4 main topics that we are going to study under biotechnology and its applications. Clear? Rest we are going to study in the very last in the next class, and I think next class we are going to finish it off in one single class. I think we will try to solve it. হবে না মনে হয়, but fine. Any questions? Come on. That's it. Just wait. That's all for now. Just wait.
আওয়াজ করতে আসলে